Hello students, let us start with the next chapter in biology, that is excretory product and their elimination. So in this chapter we will learn about the process of excretion. So what is excretion? Excretion is basically elimination of metabolic waste materials like ammonia, urea, uric acid from the body. If these materials accumulate in the body, then they can be very toxic. So it is necessary that we remove them from our body all the time. So there are various types of excretion based on the materials which are excreted by various organisms. These animals can be called aminotelic. So the type of excretion is a monotelism, ureotelism, where they excrete urea. Then there is uricotelism, where they excrete uric acid. So let us understand all this in detail. Aminotelism is when the organisms excrete ammonia. Aminotelic animals generally are aquatic invertebrates, insects, bony fishes, aquatic amphibians, etc. As I told you before, ammonia is highly toxic, so it is necessary that it is removed from the body immediately. The elimination of ammonia also needs excess water. Ammonia is readily soluble in water and is excreted by diffusion through body surfaces or gill surfaces as ammonium ions. Kidneys do not play any significant role in its removal. Next, let us understand ureotelism. So these animals excrete urea. These are called ureotelic animals. These generally include cartilaginous fishes, terrestrial and semi-aquatic amphibians, aquatic and semi-aquatic reptiles, mammals, etc. In liver, ammonia is converted into a less toxic urea, so it needs only moderate quantity of water for excretion. Some amount of urea may be retained in the kidney matrix of some animals to maintain a desired osmolarity. Next, let us understand what is uricotelism. Ureotelism and uricotelism are needed for water conservation. Otherwise, the animals will face problems. So here, uric acid is eliminated. It is generally insoluble in water, so water is not required for its excretion. These animals generally include insects, crustaceans, land snails, terrestrial reptiles, and birds. Excretory organs in animals will also be different. They can be protonephridia, that is flame cells in Platyhelminthes, rotifers, some annelids. They can be nephridia in annelids. They can be malpighian tubules in insects. They can be green glands or antennal glands in crustaceans like prawns. And in higher animals, including humans, it's generally kidney. Let us now try and understand the human excretory system. So the human excretory system includes a pair of kidney where the waste is eliminated. Then we have the ureter, the tube which carries the waste. The urine is then stored in bladder and it is removed through an opening called urethra. Kidney is the main organ for excretion in humans. It is a red-brown bean-shaped structure which is enclosed in a tough three-layered fibrous capsule. It is situated between levels of last thoracic and third lumbar vertebra. Its length is around 10 to 12 centimeters with a width of 5 to 7 centimeters and a thickness of 2 to 3 centimeters. Its average weight will be between 120 to 170 grams. On the concave side of kidney, there is an opening called hilum. You can see here, this is the hilum, right? Through here, the blood vessels, nerves, and lymphatic ducts, ureter, enter the kidney. On the concave side of kidney, as I told you, there is a hilum. So this hilum leads to a funnel-shaped cavity called renal pelvis with projections called calyces. So it is generally made up of outer cortex and inner medulla. The medulla is the middle portion of the kidney. Medulla has few conical projections called renal pyramids, right? So these are the medullar pyramids or the renal pyramids. They project into the calyces. The cortex extends in between the medullary pyramid as renal columns called columns of Bertini. So if we look at the kidney, it mainly has three parts: the cortex, the medulla, and the inner part. The main structure and the functional unit of the kidney is the nephron. Each kidney has nearly 1 million tubular nephrons in it. These nephrons have two parts. They are glomerulus and the renal tubule. So the glomerulus is a tuft of capillaries which is formed by the afferent arteriole. You can see this is the afferent arteriole and this is the efferent arteriole. The blood comes in through this arteriole, so it is called efferent arteriole, and the blood goes away from this arteriole, so this is called the efferent arteriole. Generally, the kidney begins with double-walled cup-like Bowman's capsule. So this is an important part of the nephron and it encloses the glomerulus. So this glomerulus plus this Bowman's capsule is what makes up the Malpighian body or the renal corpuscle. The next part that we will study about is the renal tubule. So this tubule continues with the PCT, that is the proximal convoluted tubule. Convoluted because you can see here it is highly twisted. Then we'll have the Henle's loop. Then we'll have the distal convoluted tubule. Henle's loop is a hairpin shaped structure. It has an ascending limb as we can see here and a descending limb around it. The DCTs of many nephron open into the collecting tube. DCT is the distal convoluted tubule which opens into the collecting duct. The collecting ducts extend from the cortex to the inner part of medulla. They converge and open into the renal pelvis through medullary pyramid in the calyces. Malpighian body, the proximal convoluted tubule and distal convoluted tubule are situated in the cortex area. The loop of Henle dips into the medulla. So this is the cortex and this is the medulla and this is where various structures are situated. The efferent arteriole emerging from the glomerulus forms a fine capillary network around the renal tubule. A minute vessel of this network runs parallel to Henle's loop, forming a U-shaped resa recta. There are types of nephrons which we are going to learn about. So first is the cortical nephrons. They form about 85% of the structure. In this, the Henle's loop is short and it extends only very little into the medulla. Vasa recta is absent or highly reduced here. Then we have the juxtamedullary nephrons which form about 15% of the structure. So here the Henle's loop is long and it runs deep into the medulla, and here we see the presence of vasa recta. Now let us understand what happens during urine formation. So urine formation consists of three main steps. They are glomerular filtration, reabsorption and tubular secretion. Let us try and understand what happens in each step. First, let us start with glomerular filtration. We studied about glomerulus earlier. So the glomerular capillary blood pressure causes filtration here in three layers. There is a high blood pressure maintained because of the structure of the glomerulus. So because of this, all the unwanted particles will be filtered out. So these three layers are endothelium of glomerulus blood vessels, epithelium of the Bowman's capsule, basement membrane between these two layers. So because of the high pressure here, the filtration will occur easily. The epithelial cells or the podocytes of the Bowman's capsule are arranged in an intricate manner, leaving some minute spaces called filtration slits or slit pores. So this is how the filtration will occur easily over here. Let us understand this process in detail. In glomerular filtration, almost all constituents of blood plasma except the proteins pass into the lumen of the Bowman's capsule. About 1100 to 1200 ml of blood is filtered by kidneys per minute. It constitutes about one-fifth of the blood that is pumped out by each ventricle of the heart in a minute. So in this way, the filtration will occur in glomerular filtration. After this filtration, almost all constituents of blood plasma except proteins pass on to the lumen of the Bowman's capsule. So the amount of glomerular filtrate formed per minute is what we called as glomerular filtration rate. Normally, this rate will be about 180 liters per day. Now, this 180 liters of glomerular filtrate is passed daily, but only 99% of this is reabsorbed, but about 99% of this is reabsorbed by the renal tubules. Because our body cannot afford to waste all the constituents which are required to maintain the osmotic balance. So, normal volume of urine released is about 1.5 liters. As I told you that our body cannot afford to waste glucose, amino acid, sodium. So these materials are reabsorbed actively. Active transport means it is going from lower concentration to higher concentration. And nitrogenous waste are absorbed passively. That means they are going from high to low. Passive reabsorption of water occurs in the initial segments of the nephron. So in this way, 180 liters of glomerular filtrate is produced daily, and as we discussed earlier, most of it will be absorbed here. So this is called selective reabsorption because we are selectively reabsorbing glucose, amino acids and sodium, and then the rest of the fluid goes away. The PCT reabsorbs most of the nutrients and 70 to 80% of electrolytes and water. Simple cuboidal brush border epithelium of PCT increases the surface area of the reabsorption. Loop of Henle maintains the high osmolarity of medullary intestinal fluid. So it is necessary to maintain this osmotic balance, otherwise the cell will not survive. The descending limb is permeable to water but almost impermeable to electrolyte. So this will concentrate the filtrate. In ascending limb, minimum reabsorption occurs as it is impermeable to water, but it allows transport of electrolytes. So here the filtrate will get diluted, and in this way reabsorption will occur. In DCT, conditional reabsorption of sodium and water will occur. The collecting duct extends from the cortex to the inner part of the medulla. So this reabsorbs large amount of water to concentrate the urine. It also allows passage of small amounts of urea into the medullary intestinium to keep up the osmolarity. It is very important that the osmolarity is maintained. So till now we finished filtration and reabsorption. In filtration, all the harmful substances were removed, and reabsorption, all the necessary substances like glucose, sodium were reabsorbed back by the body. Then we'll have during tubular secretion, the cells of PCT and DCT maintain the ionic balance, that is sodium and potassium balance, and acid-base balance to maintain the pH. Because here they selectively secrete hydrogen ion, potassium ion and NH3 into the filtrate, and they absorb HCO3 from it. So this is how tubular secretion is complete, and this completes the process of urine formation in the three steps that I told you. The first step was filtration, the second was reabsorption, and the third is tubular secretion. I hope you all understood the process of urine formation properly. In the next video, we will learn more about this chapter. Thank you.